Yick Chong Lam, Yale University, USA.
Current drugs rely heavily on either small molecules or proteins as therapeutic agents. Small molecules like erythromycin or vancomycin, also known as the drug of last resort, are used to treat bacterial infections. However, proteins, which are much larger molecules, include antibodies like Herceptin or blinatumomab, which are used to treat cancers. However, their individual, separate, widespread use has led to an exacerbation of multidrug resistance. However, solutions to combat this have been through the discovery and engineering of new molecules and proteins, but efforts are faltering. As more and more money is spent on research and development, the number of new molecular entities, in this case small molecules or proteins, has remained steady. Of these entities, about 20% are attributed to proteins. I have unified small molecules with proteins in the form of small molecule protein conjugates as an innovative class of therapeutic agents. Biologically, each part of this conjugate is synthesized through different mechanisms. Proteins are synthesized ribosomally, while small molecules are synthesized through non-ribosomal chemistry. I have united both disparate biological pathways towards these conjugates. I have engineered cells to produce these small molecule protein conjugates, all in vivo, within the bacteria themselves, without human intervention. As the bacteria replicate and divide, they can mass produce these conjugates, which we can then isolate and purify for functional testing. As a model, I have generated a conjugate in which the small molecule is a siderophore and the protein is an antimicrobial protein. When the siderophore binds iron, binds iron from the host which pathogenic bacteria need for survival and recruits the iron to the pathogenic bacteria by binding to the cellular membrane. It brings the antimicrobial protein into close proximity, which can then exert its cytotoxic effects, resulting in cell death. So as you can imagine, these conjugates can be variable. The small molecule or the protein can be interchanged with a variety of small molecules or proteins, and this allows us it gives us the potential to access diversity of conjugates that can be used to combat many diseases, breaking down the wall of multi-drug resistance while also breaking down barriers in synthetic biology. Thank you.